Hello everyone, my name is Jong-Hyuk Lee and I'm an assistant professor in the Department of Biomedical Sciences at Mercer University School of Medicine. Today I would like to talk about one of my recently published projects entitled Cockayne syndrome mice reflect human kidney disease and are defective in de novo NAD biosynthesis. Cockayne syndrome, CS, is a rare autosomal recessive accelerated aging disorder characterized by early onset neurodegeneration, vision, and hearing loss. CS patient cells show abnormally increased mitochondrial respiration, accumulation of damaged mitochondria, and impaired mitophagy. The cause of the disease mostly comes from genetic mutations in either CSA or CSB gene, which are members of transcription-coupled nucleotide excision repair. One of the molecular characteristics of CS is that it has accumulated intracellular chromatin poly-ADP ribosylation. Poly-ADP ribosyl polymerase, PARP1, is recruited to the site of DNA damage and generates PAR chains using intracellular NAD, marking these sites for DNA repair factor recruitment to facilitate DNA repair. However, without functional CSA or CSB, PARP1 may reside on DNA damage, and persistent PARylation causes intracellular NAD deprivation that leads to mitochondrial dysfunction and accelerated aging. To summarize, in Cockayne syndrome, we know that CSA or B depletion causes DNA damage accumulation, which induces PARP1 hyperactivation and chromatin poly-ADP ribosylation accumulation. Subsequently, NAD deprivation causes mitochondrial dysfunction that results in accelerated aging. So these has been cross-confirmed with many follow-up studies developing interventional approaches such as suppressing DNA damage by chromatin remodeling, preventing intracellular NAD deprivation by PARP1 inhibition or NAD supplementation, or facilitating mitochondrial biogenesis using chemical or genetic interventions. However, one of the previous studies claimed that in animal models, PARP1 inhibition does not recover all the features of CS, including mitochondrial function, while NAD supplementation has a stronger effect on improving CS. This suggests an existence of an additional pathway that leading to intracellular NAD deprivation in Cockayne syndrome. Not all of the molecular etiology of CS has been investigated, and renal pathology is one of them. Renal failure is an important feature in CS patients and has been reported since mid-1960s. However, due to extreme rarity of the disease, renal pathology in CS is largely unknown. These are the list of knowledge gaps. First, is there a way to study renal pathology in Cockayne syndrome? If so, what pathological changes occur and what are the underlying molecular mechanisms in this? Finally, are there any therapeutic strategies for prevention or cure? To answer the first question, we can think about animal models, of course. Unfortunately, not all of the human CS symptoms are found in mouse model. However, we were very lucky to find that CS model mouse showed phenotypic changes in their kidneys. The kidneys in CS mice showed noticeably small size, weight, and paleness compared to their corresponding controls. Some of them showed significantly deformed morphology, having big water-filled blisters on their surface. Also, when we measured cortical to medullary ratio, we noticed that the kidneys from CS mice had decreased cortical medullary ratio compared to wild types. If we review the structure of the kidney, cortex is the area where nephrons are located. Nephrons are functional units of kidney where blood is filtered out through glomerulus and reabsorbed through proximal and distal tubules. Because of the time limit, I'm not going to go show all the details of the data today, but pathologists blinded histopathological imaging analysis found tubular lesions in 17 to 21 weeks old younger CS animals, while older animals of 45 to 67 weeks of age showed tubular lesions as well as glomerular lesions. If you look into the renal structure again, the proximal convoluted tubules is a crucial part of the nephron in the kidney, responsible for the reabsorption and secretion of various substances. One of the characteristics of these proximal tubular epithelial cells is that they have higher number of mitochondria. This is mostly because these cells have higher energy demand for active transport of solutes that are needed for maintaining overall homeostasis and function. By picroserous red staining, we found decreased fibrosis around nephron and proximal tubules from CS mouse kidneys, indicating persistent damage and inflammation. We also found decreased volume of mean glomerular area and tubular area in CS mice kidneys, which are consistent with my earlier data showing decreased cortex volume and overall size. Tunnel assay is an experimental method staining DNA fragmentation that specifically detects cell apoptosis. When we performed tunnel with tissue sections, we found significantly increased tunnel staining positive cells in CS kidneys. With the serum and urine analysis, we found increased blood urea nitrogen and creatinine in the serum of CS mice, and decreased urea nitrogen and creatinine in their urine. These data suggest that the CS mice has impaired overall renal function compared to wild types. Also, in CS mice, we found increased kidney injury markers for serum, as well as urine samples that are indicators of significant kidney damage. To better understand the etiology of the renal pathology in CS animals, we performed RNA sequencing analysis using kidney tissues. One of the commonly changed gene ontology terms that showed up in CS animals was genes that were related to tryptophan metabolism. This was very interesting because tryptophan metabolism is a very important element in intracellular NAD homeostasis. Maintaining sufficient intracellular NAD level is crucial in maintaining renal tubular epithelial cell homeostasis because NAD is an essential cofactor for mitochondrial energy production through oxidative phosphorylation. There are well-known several different NAD synthetic pathways in mammals. First one is the price handler pathway using nicotinic acid. Second is the salvage pathway, recycling NAD from nicotinamide or nicotinamide riboside. Finally, there is de novo pathway synthesizing NAD from tryptophan. The key and rate-limiting enzyme in this pathway is called QPRT. It is known that the pathway choice among these NAD synthetic pathway is mostly tissue and cell type specific. Notably, kidney cells mostly rely on price handler and de novo pathway for NAD synthesis, and also have the highest QPRT expression among all other organs and tissues. When we looked more deep into the genes that are members of nicotinate and nicotinamide metabolism pathway, we found CS animals have greatly decreased QPRT expression. We checked the NAD levels in the kidney tissues and confirmed that the kidneys from CS animals are having decreased levels of NAD. Also, we found significantly decreased QPRT mRNA expression levels in the kidneys of CS animals. Next, we wanted to understand why QPRT expression has been decreased in CS animals. Again, please note that QPRT is the key rate-limiting enzyme in de novo NAD biosynthetic pathway. Previous study demonstrated that CSA and CSB mediates proteasomal degradation of transcription repressor ATF3 on target gene promoter. This suggests that when there are no functional CS proteins, ATF3 persistently bound to target gene promoter, thereby repressing mRNA transcription of its target gene. So we hypothesized if this could be the case with QPRT gene. In other words, when CS proteins are absent, ATF3 could repress mRNA expression of QPRT gene. First, we tested if there are any changes in ATF3 expression in CS animals. When we performed Western blot using kidney tissue samples, we found greatly increased ATF3 protein levels in CS animals. On the other hand, QPRT protein expression was greatly downregulated when ATF3 levels were high. When we then wanted to confirm if the increased ATF3 protein and decreased QPRT expressions were CSA and B dependent. For this, we knocked down CSA or B in HK2 human kidney proximal tubular epithelial cells. As you can see here, when we downregulated CSA and CSB by siRNA transfection, ATF3 expression is significantly increased, while QPRT expression is downregulated compared to control. We also confirmed intracellular NAD level being downregulated when CSA or B is depleted. By computational analysis, we found potential ATF3 binding sites along human QPRT gene locus. We evaluated the presence of ATF3 protein by chromatin immunoprecipitation, CHIP, using ATF3 specific antibody. As a result, we found evidence of ATF3 occupancy on both enhancer and gene body of human QPRT gene locus. These data suggest that CSA and B depletion stabilizes ATF3 protein, and stabilized ATF3 persists on QPRT gene locus to repress QPRT transcription. Finally, we tried rescue experiments on CSA and B knockdown HK2 cells. We used nicotinamide riboside, NR, that can bypass de novo pathway using salvage pathway as an alternative NAD source. When we depleted CSA or B by siRNA, the expression levels of kidney injury markers increased significantly. However, when we replenished cellular NAD level with nicotinamide riboside treatment, this was suppressed almost to the level of scrambled control. Additionally, when we performed tunnel staining to detect cellular apoptosis, we found NAD supplementation significantly suppressed CSA and B knockdown-induced tunnel staining positive cells. These results again suggest that CSA and B depletion induces apoptosis in kidney tubular epithelial cells by intracellular NAD deprivation. To summarize our findings, in kidney proximal tubular epithelial cells, CSA and B absence causes ATF3 stabilization on QPRT promoter, which suppresses QPRT mRNA transcription. This causes impairment of de novo NAD biosynthetic pathway, which leads to increased apoptosis. And based on our histopathological analysis, tubular injury may have been started as soon as 2 months of age in CS mice. Chronic tubular injury may have eventually developed to glomerular atrophy and sclerosis, ultimately resulting in decreased glomerular filtration and overall kidney function. With this set of experiments, I think we can now answer to these outstanding questions. First, how can we study renal abnormalities in CS? We think CS mice reflect kidney features in CS human patient and can be a good model to study this. And second, what pathological changes occur in CS kidneys? We found tubular lesions that may have developed to glomerular injury eventually. And third, what would be the underlying mechanism of these kidney features? We found NAD deprivation in CS mice kidneys, which may have stemmed from ATF3 stabilization on QPRT promoter, suppressing QPRT transcription. And finally, are there any potential therapeutic strategies on these? We believe NAD supplementation to bypass insufficient de novo NAD biosynthesis may be beneficial in preventing tubular epithelial cell death. Finally, I would like to acknowledge my colleagues and collaborators who contributed to this project. My former colleague, Dr. Komo Pekkali at Ohio State, my postdoctoral mentor and good friend, Dr. Will Bohr at NIA, and urology specialist, Dr. Moshe Levy at Georgetown University, and pathologist, Dr. Avi Rosenberg from Johns Hopkins University. This work would have not been possible without their dedication. With this, I would like to wrap up my presentation. Thank you for your attention.